Shall I start?
Yeah, you can go ahead. It looks like we've got numbers slowly climbing here.
Okay.
But take it away whenever you're ready.
Thank you. So I will start the seminar. Good afternoon, everybody. My name is Cognition. Together with our other two co-moderators, Dr. Katrina Dweck and Dr. Michael Ahmadi, and all speakers today, welcome to the webinar organized by NIC and the Data Course Steering Committee, featuring 6 speakers from our Arizona ADRC Data Core, Data Management and Statistical course. For this 90-minute webinar, each of our 6 speakers will have total 12 to 13 minutes presentation, plus 2 to 3 minutes question and answers, for total of 50 minutes. To ask questions, our audience can click on the Q&A at the bottom on your right, and write your question there. But I cannot please correct me if I'm wrong. The moderator will communicate with the speaker in selecting and answering few questions. So our first speaker today is Dr. Mike Huentelman, Professor Neurogenomics Division at the Translational Genomics Research Institute in Phoenix. And here his slide. Welcome, Mike.
Thanks, Kuwei, and hi, everyone. Thanks for joining today. So we thought it might be fun to start with a topic that has become more and more important to everyone here on the call, which is electronic-based cohort study. We actually have been doing this for 8 years now, and I'll describe to you some of our lessons learned. Obviously, this is becoming more and more important and more interesting due to the COVID pandemic, which limited many of our face-to-face studies of our human subjects. So our study can be found at this website, mindcrowd.org. We are the Mindcrowd project, and this is our homepage. As I mentioned, we've been doing this now for 8 years. We have over 225,000 participants in our study from 150 countries around the world. On the bottom here is a map of our subjects from within the United States. I've colored them here based on their urban or rural location, based on US Census zip code tracks. And at the top, I'm giving you just a brief overview of the website. It's a very simple website. It only contains 7 different pages. And below each one of the pages, I'm showing you how long people spend on the website. So important things for you to realize today, from start to finish, folks average less than 10 minutes on our website. They spend most of the time doing the paired associate learning task, which is a verbal memory task. But we do share the results back with them, and they spend about 3 minutes looking at their own results at the end of their participation. Also, we ask demographic questions before and after the cognitive tasks. We've prioritized demographic questions that happen before the task to be low anxiety questions. Okay, so oftentimes when I speak about web-based testing, a lot of folks have some questions about accessibility and disparity. So I like to mention the actual numbers here based on recent data for all of you to really think about. So I'm showing you 2 different questions here. Adults who do not use the internet on top versus households that do not have access to a vehicle on the bottom. So you can see around 7% of US adults admit to never using the internet or not using the internet frequently. This percentage is dropping across time. But down below, almost 9% of US occupied housing units report no access to a vehicle, and that's largely flatline. So this is not improving this access to a vehicle. I also want to note the cost. So to own a vehicle and operate it, it's been estimated to be around $8,000 to $9,000 per year. And access to the internet, assuming different internet prices, but also sort of the most expensive approach, which would be broadband, it's around $600 to $1,500 a year. So much more affordable than owning a vehicle. And what I think is really most important is this disparity issue. So I'm showing you the percent disparity versus the white population. And you can see with the internet, we do see a disparity. It's 3% for Black African Americans and 2% for Latinos. This means increases in the number of adults within those groups that report not using the internet. But in the bottom, I hope you can appreciate these disparities are much larger when it comes to access to a vehicle. You can see 14% for Black African Americans and 9% for Latinos. So oftentimes people think that having a study on the internet is a barrier. I hope these numbers start to convince you that there are actually some places where the internet is an improvement over asking folks to, for example, drive to your laboratory, hoping they have access to a vehicle. Now, it doesn't mean just because you're on the internet, you can successfully recruit a diverse population. We've learned that the hard way. But I do want to show you some examples of how we've improved this. So I'm showing you here 2 different plots, Hispanic Latino recruitment and non-Hispanic Black African American recruitment into Mindcrowd during 2020. So during the pandemic year last year. The black dots are the daily recruitment percentage into the website. The black line is the 2020 trend line, and the red dashed line is our historical average for these groups. So what I want to emphasize is that we've seen significant improvements in recruitment of these 2 groups in 2020, 178% increase in Latino and over 200% recruitment of Black African Americans. Now, we did this by partnering with a company that specializes in marketing and communications to these groups. So the take home really is just because you're on the internet doesn't mean you get to take advantage of the diversity that's there. You really do have to still pay attention to how you recruit into your study. That's still very important. So briefly, I wanted to show you some results to convince you that the data we're collecting is indeed accurate and robust. So I'm showing you on the left side of the screen here, the results for the reaction time test, and the right side of the panel here in rainbow colors, the results for the memory test. And you can see, as predicted across age, the reaction time is slowing from left to right with increasing age, and the memory performance on the test is decreasing from left to right with increasing age. Folks are getting less word pairs correct. But I hope you can also appreciate the widespread in the data there, which reflects the nice variation in the human population with both of these tasks. There are some things that are known to be different for verbal memory. Many of you in this field are already well aware of these. First, we should see a significant difference between sexes, and we do in our study. Men are worse at verbal memory across every single age on average. And there also should be sort of a stepwise improvement in verbal memory performance based on your educational attainment. And that's shown on the right side here. With each increasing educational milestone, we see an improvement in the average performance on that test. Okay, so one thing we have to remember though is that this is the internet and you need to pay attention to data cleaning. And I'm showing you here as an example, I'm using the visual reaction time results to show you what happens when you do appropriate data cleaning. So on the left side, we're showing with the outliers intact, and on the right side, we're showing you with the outliers removed from the SVRT results. And what I just want you to pay attention to is the bottom. So in panel C, you can see the standard deviation across age. It's really all over the map, and that reflects some poorly attentive participants, for example. But after cleaning the data, we see this very nice improvement and increase in the standard deviation of SVRT performance. This is expected. So with increasing age, you see greater variation. So I think it's really important that when you start to do internet-based studies, you pay attention to how you're cleaning the data. It's a very important part of this. So just a couple results which might be of interest here, and then I'll sort of skip ahead to keep us on time and focus on some of our forward-looking things. This is the effect on memory performance of smoking status. And what you can see here is that smokers who are in the blue line have lower memory performance on the paired associate learning task compared to non-smokers who are in gold. In these statistical examples, we've controlled for every other covariate that we collect. But what you'll notice is that this is really only statistically significant in the women smokers. So this is a sex-specific effect of this lifestyle choice on your memory performance, at least on our verbal memory task. So we find this to be extremely interesting. This has been a bit controversial in the field, but I think with our study size, which here is over 70,000 participants, we believe that smoking status is a really negative effect on verbal memory in women primarily. One of the other problems with internet-based research folks often raise is the reliance on self-report. So one of the nice things that we try and do when we look at self-report error is we introduce artificial additional self-report error and ask how robust are our results. So in this case, we have introduced increasing amounts of self-report error across the bottom, and we've assayed how robust is our finding. And we find that we still uphold this significant smoking effect in women, even when we introduce 10% additional self-report error. That's on top of what already is presumed to be self-report error in our cohort. So we think this finding is going to end up being very robust. Now, what about biological specimens? We really care a lot about getting biological specimens from our participants. So one of the other complaints or one of the other critiques we have is it's really, how do you study DNA from someone who is across the country? Well, we've actually tackled that as well. So first, I want to remind you guys that we actually found that self-report of a first-degree relative with family history of Alzheimer's disease was associated with worse memory performance, especially in the younger ages. And to follow up on this, we actually collected dried blood spots. So our participants are willing to prick their fingers, collect dried blood for us, and send it back in the to the laboratory through US mail. And through these collections, we are able to look at their genetics. So we can collect biological specimens from across the country in this fashion in a pretty cost-effective manner. And what I'm showing you here is some unpublished data suggesting that we have identified some modifiers of this family history of Alzheimer's disease effect. So I'm listing the genes out over here. I'm not going to go into the specifics. That's not really the point here. The point is to show you that we, in the carriers here in blue versus the non-carriers in red for some of these genetic findings, they seem to be a little more resistant to the negative effects of having a first-degree relative with Alzheimer's disease. So it could be that these genetic factors are somewhat protective against this effect. So just to sort of summarize here in the last 1 or 2 minutes, this is the current sort of overview of MindCrowd. We do have a way for you to recruit into MindCrowd from your existing studies, and we can share data with others. So we can recruit in the MindCrowd, we can recruit from MindCrowd into your studies, and we're happy to collaborate and do that. And we're also building out some additional tests within MindCrowd as well. I wanted to leave you with a little bit of an overview of what I think is the vision here. So part of the vision is creating these mobile laboratories. So I think there's a 3-part solution to human research. And the exciting thing about these mobile labs is you can do consent and testing on site. You can process blood samples on site. And now with some of these emerging low field MRI technologies, you can actually image brains on site with the low field MRIs. But here's really the take home message. And that is, I think we need to start thinking about, as a field, the combination of these 3 research approaches to really address all of the needs. That is an internet-based approach, an in-person study approach, and then also a mobile laboratory approach. All of these under the roof of one single project, I think is really the most powerful approach. As you can see, with the internet-based approach, without it, you struggle with appropriate sample size. With the in-person, in-laboratory approach, without that, you can't make gold standard measurements. And without the mobile laboratory approach, you're really limiting the composition of your face-to-face cohort to those people who are close to you, who can get to you. If you have a mobile lab, you can drive to them. So I just want to end by thanking the Arizona Alzheimer's Consortium for having me be a part of their team, but also the grant mentioned there, the P30 grant, and of course, the assistance of the DHS and state of Arizona in support of the Arizona Alzheimer's Consortium, which is listed there as well. Thank you.
Thank you very much, Matt. You are right in time. Question from the audience. Again, you can read your question in the Q&A. Click at the bottom right of the Zoom window. We have time to answer 2 or 3 questions, or might to answer questions.
And I'll be here during the other talks. I'm happy to answer questions in the chat box
Sure,
if that's appropriate.
Right, if you have a time after all the talks, and we can answer some general questions to any of the speakers. All right, so maybe we can move on to the second speaker, which is Mr. Senner. He's the co-director of our data core, data management statistical core. He has been working on the 20+ years in IT, very experienced in data management, healthcare, and clinical research, and also doing data mining. Welcome, Dan, for the talk. Dan, we cannot hear you.
All right, now I think you can hear me.
Yes.
Sorry about that. Good afternoon, everybody. Just wanted to share with you a little bit of the infrastructure that we've built to accommodate our multi-site ADRC in Arizona. So we currently have 5 active physically distant sites in Phoenix and Tucson. We've had a couple others that have been active over the course of the ADRC's existence, but only 5 currently active. In terms of goals, in building out this infrastructure, we wanted to be able to collect data in a consistent manner from all sites and consolidate that and upload it to NAC in one upload. Distribute errors and alerts that come from data consistency checks once we upload to NAC. Create reports for core leaders in order to monitor the health of the program. Provide a website where investigators can view the data, samples, images, and request those for potentially collaborative projects. Leverage some of the data to support our diagnostic consensus conferences, which are held every several weeks and are done by video from all the constituent sites. And lastly, to get a better handle on the time to finalization metric that NAC measures, is sort of looking at the steps internally that can impact that. So here you see an overview of the infrastructure that we're leveraging. So we use REDCap in order to collect all the UDS data at each site. And then we've written some custom software that you see here in the middle, REDCap to Relational, which leverages the REDCap API. It actually generates a SQL Server data schema and then populates it with the data. And then from this SQL database, we have some of our tools, issue tracker, the NAC files that we transmit up to NAC, and some of the web-based report and data discovery that I'll cover in a few moments. With the issue tracker, this is one of the challenges that we have with having 5 different sites, is that we have to upload as one upload, but then get the data consistency checks and errors and alerts fed out to each of the participating sites. And so we created a custom piece of software called the issue tracker that will actually parse the NAC report and properly associate each error with each site. And then they can go into the issue tracker and correct any errors or verify any alerts. And it works in kind of this circular workflow where we upload data to NAC, the error report gets loaded into the issue tracker, the errors are assigned each site, site corrects and verifies the errors are complete, and then we go ahead and re-upload them. And sometimes it takes a couple iterations before the packets get finalized. Just to show you a little bit about the issue tracker interface, we do copy down the raw error report from the NAC interface, and then that gets uploaded into this interface. So this is a log of all of the error reports that we've uploaded. And then an individual site will be able to search by a single participant ID or some of these other filters here, and they'll get a row. And then when they expand that row, they'll be able to see the details of the error. In terms of online reports, we currently have about 20, and we're continually adding these. They have the option to export to PDF, Excel, and more recently we've added the ability to people to be able to sign up for having them delivered on a monthly basis. So they're very much tabular in nature. So I didn't want to actually go through the individual ones. And then in order to make other investigators aware of the data, both internally and externally, we've created what we're calling our data discovery tool. And I'll just go through sort of the interface here real quickly. So you've got various options here, and you can refine the queries here. And then based upon the choices that are done here, you'll see various infographics here, some tables, and other items here. So you can get sort of a good feel for the cohort that we have in the ADRC here in Arizona. And then finally, if you want to, you can submit a request for data or samples, and then that goes to a committee for review and approval. One of the things that we've piloted recently is the DCC report. This just has the most recent history for a participant. So during the DCC conference, when somebody wants to refer back to a participant's last status, they can very easily do this. They just enter the PTID, and then their last visit information shows up. And then finally, in terms of what we're calling a DCC tracking report, these are kind of the internal steps. So we do record the data initially on paper, and then it goes to the DCC for review. Data is then entered in the REDCap, NAC upload, and then the time to finalization. So we find that with each of our sites, things can get hung up at any one of these steps here. So we're trying to get greater visibility into each step in the process in order to get a handle on our time to finalization, which is again a metric that NAC measures. Acknowledgements, want to acknowledge 2 NIH-funded grants, as well as the contribution of our state funding to this infrastructure. And finally, questions.
Thank you, Don. Again, also in time interview and earlier give us plenty of time to answer questions. Actually, there's one question in the Q&A need you to address, Don. The monthly report, you know, you can request, people can request to send email or email. Can that monthly report be generated dynamically, or have to be in manual process? If it go ahead.
Yeah, it can be done. You can go into it live and generate it on demand. But we just find that a lot of our investigators are generally too busy to remember yet another site and yet another password. So we're just providing this as an option so that they can subscribe to the ones that they're interested in, and it just comes to their email, and then they don't have to worry about logging into a site or anything like that.
Thank you, Don. This question was from Sarah. Sarah, if you have a follow-up question, or if you want to have additional answer, you can type in the Q&A window like you did. Okay. Any additional question? Okay, here's another one from Sarah. Can you provide some detail down on how the automatic email work?
It's just based on ASP.NET C# code that we have in a scheduled job on one of our servers, where it just queries the database that I was showing in Microsoft SQL Server, and it queries, you know, who's signed up for which report, and then generates the email and attaches the appropriate report based on the records that are stored in the SQL Server database. So that's kind of our infrastructure, is C# and .NET.
Thank you. Yeah, thank you, Sarah said. Looks like we don't have any additional questions. So that's going to be the first two speakers that I moderate. I'm going to hand this over to Mike.
All right, thank you, Kuei. I will go ahead and introduce our next speaker. Our next speaker is going to be Dr. Gene Alexander from the University of Arizona in Tucson. Dr. Alexander is the director of the Brain Imaging, Behavior, and Aging Laboratory at the University of Arizona, and he serves as the core leader for the Arizona ADRC Biomarker Core. And one of his primary research areas over the past 25 years has been the use of novel multivariate statistical models on neuroimaging data, which I believe is what he will be discussing today. So, Gene, the floor is yours.
Great. Thanks, Mike. I appreciate it. And thank you to Kuei and the others for helping to organize all of this. And hello, everyone. I'm really appreciate the opportunity to tell you a little bit about the work that I've been doing over the years, looking at multivariate kinds of techniques, and specifically looking at how structural brain imaging with MRI can help inform us about the regional patterns that are associated with aging and Alzheimer's disease. So in terms of how we approach image analysis in Alzheimer's disease and aging, typically, of course, we rely on univariate analytic methods that help us to evaluate individual regional differences in brain structure in the context of aging or an Alzheimer's disease risk. And in this case, although these are really valuable techniques that tell us about how these brain regions differ, it may not necessarily be as powerful as other techniques for detecting more subtle effects if we have to apply multiple comparison corrections over many voxels or many vertices, or even our regions of interest. It also may not fully capture the richness of our structural imaging data and how these patterns of selective regional vulnerability develop. So multivariate techniques can help us understand potentially more, or at least complementary to the univariate techniques, to help address how brain regions covary with each other, trying to capture something about the selective regional vulnerability that we see. And then also can be applied in multimodal frameworks where we can combine different data sets, can potentially enhance our ability to detect early the effects of aging and Alzheimer's disease, track its progression, and even also potentially have applications eventually for prevention and evaluating prevention of disease. I'll be talking about a method called the scaled subprofile model, one that's been around for now approximately over 2 decades, but it's really one of many multivariate methods that are currently being used and have been developed over the years. In the case of the SSM method, it really identifies network patterns of spatial covariance and differences in pattern expression. And we can use this technique across multiple types of imaging modalities. So with SSM or its variants like ordinal trend analysis, it's been applied to cerebral metabolism with positron emission tomography, functional MRI, activation studies, white matter diffusion integrity, and structural MRI are just some of the ways it's been applied over the years. And so what I hope to do today is to be able to just illustrate some of the different aspects or ways it's been applied, how these kinds of multivariate structural covariance methods can help us to understand more about brain aging and the risk for AD. And one of the ways in which I've been approaching this is really to think about how we can translate from humans in human studies to non-human animal models, with the idea that we can benefit from that extension so that we can understand some of the more molecular bases once we identify common and shared regions that are affected. So really just to get back to the fundamental question we're trying to address, if you see here a trajectory of age versus heading towards dementia, and you think about this figure where you have years on the x-axis being the age, your age as you progress, and cognitive function on the y-axis, you may be on a trajectory towards normal aging, or veer down into mild cognitive impairment and eventually dementia. And the real challenge that we all face, and in neuroimaging as well as other biomarkers, we're trying to identify people who are at risk preclinically. In this case, how do we identify people where there are subtle differences between these two groups and who's going to go on to develop it? But even more importantly, we want to be thinking about ways that we can bend back these curves, both for those going to dementia, but also those showing aging effects. And having techniques and methods to evaluate that and test those kinds of techniques or methods are really important. So the skeletal subprofile model is just one technique that I've been using for many years. And this is a technique that decomposes neuroimaging data, subject and region data, into using a modified form of principal component analysis to generate regional patterns that correspond to participant scores, network scores. It can be applied exploratory manner, or it doesn't really preclude the idea of having hypotheses and having hypothesis-driven approaches, but really focusing on meaningful variants of interest and how that variable might be expressed in the brain through the structural imaging in this case. Linear combinations are used in SSM components to test the pattern's expression, and they can be related directly to variables of interest. So in the case of age, a demographic characteristic, health status, genetic risk, disease status. In this case, because it's really focusing on a linear combined model, we don't have to rely on multiple comparison corrections because the number of statistical tests is done at the multivariate level. And additionally, we can use criterion like AIC or BIC techniques to optimize our choice of model selection. In addition, we use bootstrap resampling in its current forms, which can allow us to get confidence intervals over the individual regional contributions in the data. And these can be on the order of hundreds to tens of thousands of iterations to give us a very good representative confidence interval around the individual contributions. So how do we apply this kind of technique? Well, in terms of what kinds of findings we see with aging, over the years we've looked at it, we were really the first to apply it to the normal aging or healthy aging process in structural MRI. And this just shows an example of a study done some years ago where we looked at a small group of individuals in their 20s to 70s, and we were able to identify 52% of the variance in the expression of their age, their chronological age relative to their brain age. And this finding really shows that we can do a pretty good job of identifying a person's chronological age simply by the MRI scan. We then were able to combine this with another cohort, a separate collected cohort of individuals in their 20s to 80 years of age, and get a sort of a convergence of a common shared pattern that reflected aging, which is shown here on the right. And in this case, you can see the areas in blue in these lateral view of the brain. This is the front and this is the back of the brain. This is a medial cut through the center of the brain. And you can see these medial areas of reduced gray matter volume that are consistent with aging that we've seen in other studies. Additionally, we see small areas in normal aging where there are parietal and temporal areas involved with reductions, as well as relative preservation in cerebellum and anterior temporal lobe areas. So this is a pattern that we can build on and try to converge on a common shared aging pattern using this kind of technique. Additionally, we can look at risk for Alzheimer's disease, and we're very interested in looking at the APOE gene, the greatest or most robust susceptibility gene for Alzheimer's disease risk. But in this case, we're interested in looking at people who are young and to middle-aged. And this is really sort of an initial proof of concept type comparison where we wanted to see whether we could identify a pattern that differed between E4 carriers, the individuals who are increased risk for Alzheimer's disease, versus non-carriers. And we were able to identify a significant pattern in these young adults, young to middle-aged adults, which is decades before the onset of any cognitive or clinical symptoms. And what we see here is that they have areas in the brain that are involved or overlap with what we see in normal aging. So we have those medial aspects of the frontal cortex, we have lateral frontal cortex, and areas in the parietal and temporal regions, which were associated with the difference between the E4 carrier status and non-carrier status Of course, and there's notable variability in this cohort, and we certainly want to build on this to expand on it, but just illustrate the point that we can identify gene-based or genetic-based differences using this kind of pattern technique that may reflect a morphological vulnerability for later development of Alzheimer's disease later in life. Now, extending this into a multimodal framework, one way that we can do that is illustrated here. We're very often interested in looking at vascular lesions in the brain with white matter hyperintensities, not just gray matter volume. And this just illustrates the kind of MRI scan that where you can get a volume of these areas that are periventricular or deep white matter lesions reflecting small vascular injury. And we can segment those and identify a total volume in the brain that reflects the amount of this white matter hyperintensity volume that each individual has. But the question I had was whether we can look at how we can see how this is related to gray matter cortical volume. And so through the collaboration with colleagues at Columbia University as part of the North Manhattan Aging Study, we're able to get very closely matched individuals who are normotensive, controlled hypertensive, and non-controlled hypertensive individuals. And we were able to identify a pattern where the white matter hyperintensity volume was expressed in terms of cortical regions of the brain in volume on a voxel basis using this kind of morphometry technique. In this pattern, when we look at those individuals in the normotensive group, the hypertensive or non-controlled, we see a significant relationship with the pattern expression is higher in those who are uncontrolled and hypertensive compared to controlled hypertensives and compared to relative to non-hypertensive individuals. Again, pointing to the idea that we can have these patterns as potential biomarkers of vascular damage reflecting cortical effects in the brain that could potentially be looked at in terms of how it could exacerbate vascular injury, vascular dementia risk, or Alzheimer's dementia risk. So looking at those kinds of effects in aging and risk with genetics, and risk with vascular injury or damage in multimodal frameworks, then want to extend this and illustrate how we can extend this into non-human animal models. And then working in the rhesus macaque model of aging, we were able to demonstrate that in a small group of old versus young rhesus macaques with MRI scans gathered on these participants, we were able to apply the same techniques that we used with the SSM analysis in human studies in the animal model framework. And we were able to identify a pattern of significant reductions in areas of the frontal and anterior temporal lobes, as well as some relative preservation in basal ganglia. And this pattern was different between the old versus young animals. And furthermore, we see that when we look at delayed response performance, this was data analyzed and performed by Peter Rapp as a collaborator on this study, and also the study was done with Carol Barnes here at the University of Arizona. We were able to see how higher levels of performance, or say poorer levels of performance on this task was associated with a higher expression of this pattern across the two groups. Interestingly, we also see variability in the monkeys in terms of their performance. We see some older monkeys who perform more like young, and some younger monkeys that perform more like old, and really brings up the question of what kinds of characteristics do we see that are different in these kinds of variability. And now extending this more recently, we've also been able to look at this in the context of a rodent model. This is with Fisher 344 rats. We had 10 old and 10 young. We were able to investigate how, using the same approaches we've walked through in normal aging and genetic risk and vascular regions and in the non-human primate now into the rodent model, we can begin to test patterns that we can differentially discriminate between these age groups in terms of their pattern. And we see, again, frontal areas of the rat as being decreased, as well as perirhinal and cerebellar regions bilaterally, and also some areas of relative preservation and somatosensory and thalamic areas, midbrain, and small selective areas in hippocampus. And then furthermore, we were able to see how in maze learning with the Morris water maze, poor performance on this task is associated with a higher expression of this pattern. So again, showing just like in the monkey that we can see behavioral relevance to these patterns and that they also are associated in key differences. And I think that's an important distinction in terms of how this method identifies meaningful patterns that could be useful going forward. So what I hope to try to illustrate in this very brief presentation is that structural covariance analyses with SSM can identify regional patterns of brain morphology in brain aging and identify group invariant shared regional features. That the pattern can be associated with key variables of interest like aging and Alzheimer's risk. We can apply it in a multimodal framework And the network patterns can be then extended to preclinical translational studies, with the opportunity then, by identifying key patterns and regions, we can go more deeper into molecular studies in those cases, those models. And then finally, that these kinds of multivariate techniques, I hope you can see, it can be very powerful and useful in trying to characterize neuroimaging biomarkers in this context of brain aging and AD risk, and that potential for helping detection, tracking, and prevention therapies. And finally, I just wanted to acknowledge all the collaborators that I work with on a regular basis, but also especially the Brain Imaging Behavior and Aging Lab and my team, as well as especially note Chris Haybeck and Jim Moeller, close colleagues for many years on the applications of multivariate techniques. And then I'd like to acknowledge the support from NIA, the Arizona Alzheimer's Consortium, and State of Arizona, as well as the McKnight Brain Research Foundation. And I'll stop there at this point.
All right, thank you, Dr. Alexander. We do have a few minutes to take any questions that you may have for him. And again, you can just type those into the Q&A section located at the bottom of the screen.
If there's nothing queued up, do you mind if I ask a question?
Go ahead, Ben.
Thank you, Gene. Fantastic talk. I was wondering what you think about the potential for blending the, I guess particularly the rat imaging study that you showed, with different sorts of molecular profiling from those same animals. Like, I guess I'm thinking like spatial transcriptomics or something like that. Do you think that's feasible, and do you think that would be informative to be able to connect the molecular networks with those kind of regional network findings that you were seeing?
Yeah, absolutely. I mean, I think that's a terrific question, and that really was in a way, part of the motivation for understanding whether we could translate from the human network studies into the rodent models, because really obviously we can start to look at a much finer grained molecular level that we simply can't do in human studies easily. So it's really about trying to make that jump and being able to then map areas of the brain, identify key covariance patterns that we can then identify those regions as key areas where we can or other kinds of other metrics. So I think integrating those two in a multimodal in a multimodal way outside of the imagery frame is really a terrific goal. And I think that would be something definitely to work on.
Thank you.
That's a great area we can work together. Multimodal network.
Right.
Absolutely. Yeah.
Any other questions? We have a couple of minutes. And again, if you do have questions later for the speakers, you can enter them into the Q&A function, and they will be able to address those directly to you if you have them and think of them later. We'll go ahead and move on to our next speaker, which will be Dr. Yi Su. Dr. Su is a senior scientist and director of the Computational Image Analysis Program at the Banner Alzheimer's Institute in Phoenix, Arizona. He is also the associate director of the Biomarker Core for the Arizona ADRC, and also serves on the Data Management and Statistics Core program. And one of his areas of focus for research is harmonizing the harmonization of amyloid burden across various PET tracers. So Yi, I will pass the baton to you for your presentation.
Thanks, Mike, for the introduction. I really appreciate the opportunity to talk here today. Today, I'm going to share a little bit of our recent work on trying to improve the harmonization of amyloid PET imaging data using statistical and deep learning techniques. So we have been doing amyloid PET imaging for almost 2 decades by now, with Pittsburgh compound as the initial human amyloid imaging tracer, and later on a number of F18 labeled tracer has been developed along the way. And using this figure on the right, I just want to show you that even though all these tracers are designed specifically for amyloid PET imaging, their visual appearance can be quite different. As you can see here, I have an amyloid negative example on the left and an amyloid positive subject on the right. And both for beta peer imaging and PET were done in the same person in a short period of time. And as you can see that the visual look is quite different. The main difference is that the fluid beta pair has a lot more white matter uptake as compared to PEP imaging here. In addition to these visual differences, the quantitatively derived amyloid burden measures also differs quite a bit among these different tracers. So recognizing the lack of standardization for amyloid PET imaging, a panel of experts in the field formed the celluloid working group, tried to develop a protocol to establish a standard scale to quantitatively analyze amyloid PET data. And in their paper published in 2015, they outlined several problems with the lack of standardization, including the value range are quite different from one tracer to another, or different quantification pipelines. Also, there's a lack of standard definition of the positivity cutoff. There are also difficulties in comparing data across studies, and also difficulties in interpret the findings in terms of longitudinal changes. So in their paper, they define the goal as to provide a standard protocol to analyze PET data, and then a method to scale any non-standard method of PET analysis and or any other tracers to convert that to the celluloid scale. And here I'm going to use an example to show you what the celluloid conversion does. So the conversion actually is a linear transformation defined based on a reference data set that you apply to your favorite measurement of amyloid burden measure derived from PET imaging data. And on the left, what I'm showing here is a comparison of fluid beta pair derived mean cortical SUVRs against the PET derived mean cortical SUVR measures in the same subject and before celluloid conversion. You can see that the regression line is not quite the identity line, and the range of MCSUVR distribution is quite different between the two tracers. And on the right, after the celluloid conversion, you can see that the regression line is almost perfectly aligned with the identity line across the 45-degree angles. But I want to point out that if you look at the R-square numbers, they're exactly the same. The celluloid conversion doesn't do anything to that. So then the remaining issue after we have taken the celluloid approach, which has been widely adopted by the field, include the fact that there are the shared variance between tracers to a more degree and to a lesser degree, dependent on the imaging protocols, is that the shared variance sometimes are only moderate at 70%, as reported by some of the studies cited here. And also there are differences in the variability of amyloid burden measures in the amyloid negative controls, which reflect the noises in your measurements. And it differs quite a bit among these different tracers, and also to a lesser degree, depending on your quantification pipelines. And related to this, if you define an amyloid positivity threshold based on these measures, as reported by a number of papers in the field, the cutoff is also different. And even though these is to some degree resulted of the differences in the cohort that people use to define these cutoffs, but I would argue that even if you had a perfect data set that had all these different tracers imaged for each of the individuals and using your favorite methods to define the threshold, even after cellular conversion, the cutoff would still be different. And as we also have reported earlier, that the differences in tracers also result in the differences in their power to detect longitudinal changes and the ability to, in clinical trials, to look at the treatment effects. So it is our hypothesis that these differences in the appearance of amyloid PET images and the associated quantitative measures, they're not just purely noise, they are actually tractable characteristics of these tracers and can be accounted for if we adopt a statistical or deep learning approaches to try to characterize these differences carefully and try to model that. And then we can obtain a better harmonization model. In preliminary work, we try to develop some techniques that we can use to improve the harmonization. And we want to demonstrate that these models are generalizable to independent data set. And also they show real world benefits. So here's the data set that we are looking at. So one of the data is from the Washington University in St. Louis group. They published the OASIS data set, and a subset of that has 92 subjects with a head-to-head comparison for beta peer and PIP data. And another data set is hosted at the GAME website, published by the AVID group, who published the original paper that calibrate the FBP for beta peer PET amyloid PET celluloid conversion to the cellular scale. So, and the method we are adopted so far is a deep learning approach using the residual inception encoder-decoder network. In this approach, as illustrated here, it has 9 different blocks. Each are composed of several layers of convolutional neural network. And this is a technique that's developed by our collaborators at Arizona State University. And the model we adopted, the method we adopted to train this deep learning model is we use the OASIS data as a training set and the GAIN data set as the independent test set. And we did a tenfold cross-validation. And right now we're implementing that as a 2D model to operate on the image slices. And all the data has been pre-processed and converted to SUVR images and transformed into the standard space. And we used the flow beta pair slices as the input, and the deep learning model generates an imputed PIP image based on each FBP slice. And we assess the model performance by looking at the main cortical SUVR measures and also the voxel-wise spatial correlation. So here is what we found. We are able to significantly improve the global amyloid burden measures, as you can see on top here in the OASIS dataset. And this improvement is highly significant. And also we demonstrate that using the deep learning model trained on the OASIS dataset and applied to the independent GAINs dataset, we also obtained significant improvement in the agreement of global amyloid burden measure. And if we look at the data visually, you can appreciate the much improved visual similarity between the imputed PIP images from flow beta pair data with PIP. And also when we look at the voxel-wise spatial correlation, we also detected a statistically significant improvement. Again, on top, we're looking at the OASIS data, which we use the tenfold cross-validation. And down here is an independent data set that's completely separate from the training data. So is this kind of improvement useful? So we look at the amyloid positivity cutoffs derived based on this data, based on this model, and we can see that if we use the young control subject in the gain data set and estimate their variance in the young control group and define the 95% specificity cutoff from the PIP data, the cutoff is at 8.2 cellular unit, and for the flow beta pair, you would get 16.1 because of the higher variance in the young control group. And after applying the harmonization model, we substantially reduced the variance in the young control group, and we obtain a positivity cutoff that is much better in line with the real PIP data as compared to the flow beta pair images. So in summary, we propose a deep learning based approach to harmonize amyloid PET data at a voxel level. We demonstrate that it significantly improved agreement in both global amyloid burden measure and voxel-wide spatial correlation. And this is further confirmed. We further confirm the generalizability using separate independent data set. And also we saw real world improvement in terms of amyloid positivity cutoff definition. So there's still quite a bit of work to be done. So we're going to further improve our model, including try to see if we can take advantage of the MR data that's available, and also explore 3D models. And also we will be exploring the generalizability of this approach for tau PET data harmonization. I'd like to thank the wonderful team at the Banner Alzheimer Institute, and also my collaborators at Arizona State University, University of Pittsburgh, and Washington University in St. Louis. With these 4 different institutions, we're currently pursuing an R01 application. And also we have worked with folks at APOE and the University of Wisconsin for their contribution of data set for this heavily data intensive project. And now I'm happy to answer any question you might have.
Thank you, Dr. Su. It was a very great presentation. We do have one question that has come in already. The question is, for the PET harmonization, what would you use as your training data set? Or I guess another way to frame this would be, what would be the source of the data for the training data set?
So ideally, we would want to have head-to-head comparison data that's acquired in the same subject with different tracers over a short period of time. So far we have collected some data set like that. Like I mentioned, one of that is from WashU, and the other from the AVID group published at the GAIN website. And in the future, so in our R01 grant, we are actually proposing to acquire some more data to facilitate that effort.
I guess just to follow up on that, do you feel like that the training data set should be completely independent of the test data set? So I know that sometimes it's possible where you take a subset of your data set of interest and use that as the training data set. Do you think that using an independent data set for training is preferable?
So the main reason is to demonstrate the generalizability of this approach, especially when you consider that there might be other factors that affect the difference between the 2 different tracers. There might be differences in acquisition protocol. There might be differences in the quantification pipeline. So ideally, we would want a completely separate data set to demonstrate the generalizability. After that, once we are confident that our model is working, then we would want to train the overall model on the largest data set that's available.
Thank you. Right, any other questions for Dr. Su? They can be entered into the Q&A section.
Okay.
Right, I will hand over the floor to our next moderator, Katrina Devick, who is a biostatistician from Mayo Clinic in Phoenix. Katrina, it is all yours.
Thank you. I'm happy to introduce Dr. Ben Reedhead, who is a foundation professor and Edson Endowed Professor of Dementia Research at Arizona State University. One of his research focuses is to use multiscale network models to understand causal relationships and to prioritize therapies with experimental validation. Welcome, Dr. Reedhead.
Thanks so much, Dr. Devick. And thanks so much, everyone, joining us today for your time and attention. I'm excited to tell you a little bit about some of the work that we're doing, trying to apply different sorts of, let's see if I can advance my slides, trying to apply different sorts of integrative multi-omic data processing methods to these kind of emerging kind of large multi-omic data repositories, in particular to explore and illuminate different aspects of mitochondrial dysfunction in neurodegenerative disease. And so the spirit of this talk isn't so much to be kind of a self-contained survey of mitochondrial dysfunction, but just to hopefully illustrate some interesting methods and opportunities in what is very much an ongoing work for us. So, there's a kind of a rich and complex literature of mitochondrial dysfunction in different aspects of neurodegenerative disease. So we know that there's dysregulation of cellular bioenergetics. This is really a common feature across many neurodegenerative diseases, particularly Alzheimer's disease. We of course see signs of regional hypermetabolism in neuroimaging studies. We see shifts towards uptake of energy sources such as amino acids and fatty acids, upregulation of oxidative OXPHOS pathways and ROS productions. We have reports of reduced intact mitochondria and AD affected brains, reduced copy number, impaired base excision, and from some of my colleagues here in Arizona, findings of APOE isoform specific correlations between mitochondrial proteins with cognitive function. So there really is a complex literature that kind of sets up the motivation for why we'd want to investigate this. And so that's the motivation. And where I think one of the opportunities is some of these really massive multi-omic consortium level molecular profiling efforts. And the one I want to reference here is the Accelerating Medicines and Partnership in Alzheimer's Disease, or AMP-AD, which is this pre-competitive consortium level partnership between government, industry, academic, and nonprofit partners. And one of the things that it provides, amongst other things, is these very rich multi-omic characterizations from phenotyped human postmortem cohorts with and without Alzheimer's disease. And there's a real focus on open science and open methods. And of interest to this particular project, I want to tell you about what they have is a large number of whole genome sequences that have been generated across multiple different cohorts, four different cohorts actually. The ROS, the MAP, the Mount Sinai Brain Bank, and the Mayo cohort. And these whole genome sequences have really been generated from a range of different tissue types, collected mostly, I guess in a somewhat opportunistic way, because they were profiled really to profile germline variants. But we think there's ways this can be leveraged to profile interesting mitochondrial variables. And so, we have samples coming from blood samples, and then a range of brain tissue regions, particularly dorsolateral prefrontal cortex, posterior cingulate, and cerebellum, as well as a few others. And so our question going into this was, is there a way where we can use some of this available whole genome sequences to estimate interesting mitochondrial variables within these cohorts? And to the extent that we can, then we perhaps have an opportunity to link those variables with clinical and neuropathological and other molecular traits that we know about these same people. And so the short answer is yes, it appears we can. And some of the variables we're interested in looking at are straightforward stuff, initially, like mitochondrial DNA variants, except kind of called in a more reliable manner than what kind of the standard variant calling pipelines often do. You know, there's quite a lot of homology between, you know, stretches of mitochondrial genome and the nuclear genome, and so, you know, there's pitfalls there. But beyond looking at DNA variants, one of the other things we're interested in looking at is mitochondrial heteroplasmy, which is, you know, the detection of multiple alleles at a particular site within a given sample. So essentially, someone having, you know, more than one copy of their mitochondrial genome, more than one different copy of the mitochondrial genome within a sample. So that may be heteroplasmy even within a cell, or between cells, but within the same sample. And so there's all sorts of interesting computational challenges, which we can get into in the questions if anyone's interested, but, you know, which have mostly been solved through the efforts of others, like the Broad Institute. But let me just give you a little bit of a survey as to, you know, how some of these findings are playing out. So what you see here is a series of box plots looking at the estimated mitochondrial copy number. So this is something we're able to estimate from the whole genome sequences by looking at the ratio of the coverage between mitochondrial genome as a ratio of the, you know, the average coverage across the autosomes. And from that, we can kind of get this pseudo estimate of mitochondrial copy number. And, you know, what we see across these different tissue and cohort combinations that you see along the x-axis, you know, are the different estimated copy number. And, you know, there's just some high-level observations. You know, we see much higher estimated mitochondrial copy numbers in cortically derived samples, more so than cerebellar samples, and more so again than blood-derived samples. And, you know, this perhaps isn't a surprise to many of us on the call, you know, given what it is we know about, you know, the bioenergetic requirements of the cortex, particularly, but, you know, the brain in general compared to other peripheral tissues. What we can do, though, is kind of zoom in on one of those tissue subsets within a cohort, and showing here the ROSMAP subjects, which is a longitudinal cohort coming out of Rush University, and in particular the dorsolateral prefrontal cortex. And then, you know, we're stratifying our estimated mitochondrial copy number by disease diagnosis. And again, probably not surprising to many on the call to see that, you know, there's this fairly stark decrease in mitochondrial copy number between cases and controls, even when correcting for, you know, demographic and nuisance covariates. But, you know, one of the things we might be interested in asking as a follow-on question to this, you know, could this be, you know, simply an artifact of neuronal loss that we also know is, you know, a very early feature in AD? We know that neurons are jam-packed with mitochondria, you know, tens to perhaps hundreds of thousands, you know, within certain sorts of neurons. So, you know, perhaps this observation of a reduction in mitochondrial copy number could be simply a proxy for that more general, you know, phenomena of neuronal loss. So, you know, what's fortunate is that we have RNA-seq from these same subjects, from these same brain regions, where we can actually, you know, look at the gene expression profile, and we can deconvolve that into cell type estimates, you know, comparing it with reference panels of single cell RNA-seq data, and essentially come up with sample level estimates for these different cell type fractions. And, you know, in that comparison, we certainly see a reduction in neuronal fraction in cases compared with controls. So it could be that, you know, this, you know, that neuronal loss is actually, you know, could account for the mitochondrial copy number reduction. So, you know, we take these cell type estimates, we include it in the linear modeling that we use for you know, for this analysis, and what we see is that still there is actually a significant component of mitochondrial copy number loss in AD that's actually not explained by neuronal loss, which is interesting. And so, you know, at the moment we're exploring different ways to connect, you know, these copy number estimates with, you know, molecular and proteomic network modeling approaches to try and understand, you know, what might be some of the upstream drivers and kind of downstream response networks, you know, that could help us contextualize, you know, what's going on here. One of the threads that we are exploring is looking at mitochondrial heteroplasmy as a potential contributor here. So, you know, what I'm showing here is the number of detected heteroplasmic sites per sample. So every dot here is a sample, and we have one sample per patient. So, you know, really it's an individual's heteroplasmy within that tissue. And I guess the high level finding here is that we see much more heteroplasmy in the blood-derived samples, which perhaps isn't such a surprise, given the higher lifetime division rate of these hemopoietic progenitor cells. They've had many more opportunities than the typical brain cell to accumulate heteroplasmic variation. But if we were to zoom in just on the brain-derived whole genome sequence samples, there it's a more kind of level playing field. What we see generally is the highest level of heteroplasmy in cortically derived samples. A median, the median subject, we're detecting two heteroplasmic sites. And that tends to be lower in the cerebellar-derived samples, and it tends to be consistent across cohorts. One of the things we can ask is, of this heteroplasmy, what is the distribution around the mitochondrial genome? And what we see in these kind of right-sided, these four smaller circles on the right-hand side, we see that the heteroplasmy detected in blood samples really is distributed around the entire mitochondrial genome. And this is contrasted with what we see with the brain-derived samples, where really we're seeing the heteroplasmy restricted to a fairly narrow region of about 500 to 800 base pairs, set right around the breakpoint. And this is a region called the mitochondrial control region, which is the longest stretch of non-coding mitochondrial DNA. It's highly polymorphic, hypervariable. It's the origin of DNA replication for the light strand, and it's the origin of RNA transcription for both strands. And different haplotypes within this control region are associated with all sorts of interesting phenotypes like athletic ability and cancer, and also Alzheimer's disease. So there was this study by first author Koskin coming out almost 2 decades ago, where they reported that AD brains had an average 63% increase in heteroplasmic mitochondrial control region mutations, and that this had an interesting acceleration and interaction with advanced age. What was really interesting here was that they were able to relate these control region heteroplasmy variants with an impaired ability for those mitochondria to replicate and to transcribe in a normal fashion. So we're exploring whether or not this bears out in this current data set. Now, we're not sure yet. We think we see signs that maybe it is, but it's certainly one of the avenues that we're chasing down. One of the complementary analyses we could think about doing here is then seeing whether or not there are any inherited germline variants within the mitochondria which actually predispose to accumulating heteroplasmic variation in these control regions in the brain later in life. And doing essentially a cis heteroplasmy QTL analysis. And we actually find quite a few common variants where the minor allele is associated with an increased probability of accumulating heteroplasmic variation in the brain later in life. And we're digging into some of these at the moment, and one of the ones that's caught our eye is this particular common variant at position 10398. And the reason why it caught our eye is that when we look across the entire mitochondrial genome, this is the variant that is associated with the most independent heteroplasmy sites in that cis heteroplasmy QTL analysis. So, so the minor allele predisposes to heteroplasmy at 5 independent sites. And what's also interesting about this is when we also look across the whole mitochondrial genome, this is the variant that is actually associated with the most, with the largest number of unique phenotypes in MitoMap, which is something like the GWAS catalog for mitochondrial genetic association. So it has interesting associations with Parkinson's disease, Alzheimer's disease, metabolic syndrome, ADHD, a range of really, the implication is it's quite a kind of a pleiotropic locus. And this variant sits within the first exon of the MTND3 gene, which is the the third subunit of the cytochrome oxidase complex. And what we see in the dorsolateral prefrontal cortex is that this variant is actually a cis EQTL for this gene that it sits within the MTND3. So the minor allele is associated with lower expression of ND3. And look, just in the interest of time, I'm going to skip forward over some network modeling that we've done relating these things together. Just the 10,000-foot view is when we map out kind of the RNA and the proteomic network context of of this gene conditioned on that variant. You know, it's enriched for some really interesting AD relevant and neurodegenerative relevant nodes. But perhaps I can just say a couple of words about one of the avenues that we're taking to better model the effect of this variant and others like it using the Personal Genome Project. And this is driven by some collaborators, Dr. Rigel Chan and Dr. Elaine Lim, who helped set up the PGP in George Church's lab, which is really a resource that allows people to connect together in an open access way, whole genome data with cell lines and phenotypes. And so what we're able to do is use this resource to go in and sub-select a set of cell lines that carry and don't carry the minor and the major allele at this locus, and then do some RNA-seq and then kind of look at what the transcriptomic difference is. And this is still ongoing analysis, but what we see in these lymphoblastoid cell lines that are derived from these patients with and without this allele is that this variant really does seem to be a QTL locus for an expression QTL locus for several mitochondrial genes, most strongly for MTND4, which is actually the gene downstream of the one that we saw implicated in the dorsolateral prefrontal cortex. So ND4 is about 300 base pairs downstream of this variant. So it seems likely that there's some interesting cell type heterogeneity in these cis-QTL relationships that we're trying to understand at the moment. In the interest of time, I'll just jump forward through what our plans are, and just to wrap up very briefly. Look, I hope I leave you with a sense that that there's, I think there's a lot of exciting opportunities to blend together different integrative computational approaches applied to kind of ever increasing amounts of omics data. And what makes this really exciting is this omics data is connected to increasingly well phenotyped patient populations. So ultimately the goal here is for us to be able to use these observations to be able to better illuminate relationships with clinical and neuropathological aspects of disease. And I'm really grateful to my friends and colleagues within Arizona as well as beyond, without whose data and insight and expertise I wouldn't be able to work on this. I'm really grateful to the Arizona Alzheimer's Consortium and the Department of Health Services and the Alzheimer's Disease Corps and the National Institute of Health for their generous support of this research. And thanks so much for your time and attention.
Thank you, Dr. Reedhead. Our next and last speaker is Dr. Ignacio Perez. He is a research assistant professor at the Translational Genomics Research Institute. His research focus has been entirely on bioinformatic analysis approaches to multi-omic data for the next generation sequencing and microarray, including whole genome and exome sequencing, as well as RNA sequencing and DNA methylation. Thank you.
I don't know if you can see the presentation. I can't actually. Oops.
We can now see the shared screen.
You can see the presentation.
Only the desktop, not the
Okay.
presentation.
Oh, weird. Now, yes.
Yes.
Okay, sorry. Thank you for the introduction, Dr. Devick. So this presentation will be a quick overview about Genome-Wide and Transcriptome-Wide Association Studies. So basically, we start from a simple definition. A GWAS identifies single nucleotide polymorphism, which allele frequency vary as a function of phenotypic trait values. So we need SNPs to run a GWAS. Of course, the term genome-wide, it means we need several of these. Usually GWAS is conducted using also millions of variation. And a SNP is a variation of a single base pair in the DNA sequence. And also we need to a phenotypic trait. So it can be a disease, for example, as diabetes, or a quantitative measure variable as the body mass index. Let's see the two typical study design. We have the case control study design, and the question we need to answer is whether the allele frequency are different between cases and controls. So let's take a simple example. We have an hypothetical SNP here with 3 genotypes. We have a group of patients and controls, and we have the genotypes for this group of patients and controls. From this figure, we can see that the allele A is more frequent in the patients. So we can conclude, of course, oversimplifying all the workflow, that the SNP1 is associated with diabetes. The second study design is the QTL, quantitative trait loci. So the question we need to answer here is whether the genotypes are associated with low or high level of the variable. We have still our hypothetical SNPs, and we have a quantitative variable. Here I took the BMI as an example, and from the box plot we can see that allele C is associated with higher levels of BMI. So our conclusion is that the SNP1 is associated with the BMI. This is the typical analytical workflow. So we need a participant, can be, of course, people or animals or whatever species we are interested to study. We need to take the biological samples. Usually it's blood or saliva, but we can use also other tissues, and we need to extract the DNA. We need to collect the data so we can do that. So basically, we need to characterize the SNP all over the genome. For a typical GWAS, usually we characterize more than 1 million of SNPs. Can also be several million. So we can do this by genome sequencing, so next generation sequencing, or also by microarrays. We collect the data, and this is an important step. So we need to conduct quality control. So we need to remove low quality SNPs and samples in order to decrease the chance of false negatives, false positives. And then we run the association analysis with usually regression methods. Finally, there is the step 5. It's also quite important because we need to check our result if they are reliable. So if they are not, we need to come back to the quality control step and remove maybe additional SNP or samples, or check the data collection step. So if everything is fine, we can visualize our result and interpret the result, basically assign the significant genes we found to some biological pathway. Let's see quickly, assuming we already have collected the samples, we collected the data. So we are at the quality control step. So we have this data set with thousands of individuals for millions of SNPs. So we need, first of all, to remove samples that show the sex discrepancy. Basically, we infer the sex of the participant using the genetic variant in the X chromosome. And if the sex inferred doesn't match with the sex recorded, we can remove those samples because probably there was some mix-up during the collection or during the data collection. Also, we can compute the heterozygosity. So high level of heterozygosity can indicate a sample contamination. Low level of heterozygosity we can indicate inbreeding. So also in this case, it's better remove the samples. I forgot to tell you that usually most of the quality controls, they are conducted with the software Blink, that it's a software specialized for association studies and able to manage very large data sets. Other steps, we need sometimes to remove related samples because they can create some bias in the association analysis. For example, if we have in the group of patients related samples, there will be an overrepresentation of some genetic variants. So when we will compare the patients with the controls, we can find some significant signals, but there is not an association with the disease, but it's because the overrepresentation of those genetic variants in the patients because of the related samples. And we need to account for the population stratification. Also, this is a source of bias in the results. So for this analysis, we can run a PCA, a principal component analysis, and I recommend to use the Smart PCA software for the Hagensoft, so that has a nice and quick workflow for exclude the outliers in an automatic way without doing too many steps. We also need to remove problematic samples or problematic SNPs, for example, a sample with a low genotype rate or a SNP with a low genotype rate. And finally, usually we remove the SNPs that have a very low allele frequency, usually rare variants with minor allele frequency less than 5% or sometimes 1%, because they are not informative during the association step, because we need, they are usually underpowered to detect significant associations. And finally, also we need to remove variants that are not in our Weinberg equilibrium according to this equation. The biallelic SNP should have theoretically unexpected distribution between the three genotypes. If they do not follow the distribution, there might be some genotyping errors. For the patient, the cases group, sometimes a departure from the Hardy-Weinberg equilibrium is because of true association. So we need to be careful before to get rid of those names. Let's see what are the statistics we apply. For the case control design, we simply run a logistic regression. Here there is the command as implemented in Link. So we have the binary phenotype as a dependent variable, the genotype as a predictor, and the confounding factors, maybe the principal components for the population stratification or the sex or the batch. Of course, we need to repeat this linear regression for hundreds of thousands or millions of times each one regression for each SNP. For the QTL design, we run a linear regression, and we know that in the linear regression model, instead of the binary dependent variable, we have a continuous phenotype. And also here we have the genotypes as a predictor and the confounding factors, and we can select different model, the additive, the dominant, or recessive, but let's skip this. This is a regression approach that's currently is more used. It's the mixed linear model. This approach basically solves the issue of the relatedness and the population stratification bias. It has an increased power and it can be used for both case control and QTL. So this is the equation. We still have the phenotype that can be binary or quantitative, the covariates. We have the SNP as a fixed effect. But importantly, let's just remember about the relatedness matrix that we estimate by the genotypic data in the preliminary step. And there are a couple of software we can use for this analysis. After we got our result, we need to adjust for multiple testing. Of course, running on several hundred of thousands of tests, we have a high probability to have a larger number of false positives. So we need to adjust for multiple testing. And usually in GWAS, we can, there is this cutoff that is basically widely accepted as significance for to declare a SNP statistically significant. But as alternative approach, we can also estimate the number of independent SNPs and adjust with the Bonferroni statistic the results. This is an example of the summary statistics we obtain. We have these huge files with millions of rows with the variant T chromosome, base pair location, the effect allele, and especially the odds ratio that indicates the effect of the effect allele. If larger than 1, the effect allele is a risk factor associated with the disease or the phenotype. If the odd ratio is less than 1, the effect allele is protective, and the p-value, these are the unadjusted p-value, but if we consider the cutoff I mentioned before, only the first one will be statistically significant. This is another important step. So after we have our summary statistics, our results, we need to see if our results are reliable. So in other words, if there are too many false positives. So basically we compare our p-value, the p-value we obtain in the analysis versus the theoretical distribution, assuming not association. So for example, we expect that 1% of the p-value will be under 0.01. So we assume that most of the GWAS p-value should follow the theoretical distribution. So we will draw a plot and we can check. I will show you after this slide, and we can see if there is how we can quantify visually how much bias. And this is the second method that basically is related, because also in this case, we instead to use the p-value, we use the test statistic. So we compare the observed distribution versus the expected distribution. So this inflation factor should be closer to 1 to demonstrate the absence of bias. This is an example of QQ plot. We see the expected distribution, it's the black line. So in the blue distribution, we see it's very far from the expected distribution. In fact, the lambda, it's far from 1, it's 1.3. The magenta distribution, it's instead closer to the expected distribution, and accordingly, the lambda is 1.02. Usually, unaccepted value of lambda should be smaller than 1.1, also 1.05, it's better. So if we have high values of inflation, probably we need to remove outliers or do a different SNP filtering. This is final, the result visualization. We used the Manhattan plot. So we have the chromosome in the X, and in the Y, we have the minus log p-value, and each dot represent a genetic variant. And the red line is the genome-wide significant. So in this GWAS, they were able to identify several variants in several genes, but I think the sample size was probably 20, 30,000 samples, so we need usually a very large number. So let's talk about TWAS. The Transcriptome-Wide Association Studies integrate GWAS and gene expression data sets to identify gene trait association. One thing to highlight is that we don't need to collect new data. We basically use only the GWAS data. We can use the summary statistics, or we can use also the raw data. What we need is a reference data set where we have an independent data set. So we run a GWAS and we go on some database, and we need to find some reference data set where there are SNPs and RNA expression values collected from the same sample. For example, GTX is the most widely used for this purpose. And then we predict the expression values in our GWAS using the reference data set. And then we run the association analysis using the predicted expression values. So basically, it's only one in silico workflow. So we don't need additional experiments. Oops. So this is basic examples. We have our SNP patients controls. So we see that we have a higher frequency of the C allele in patients. So we go to check in the reference data set, and we see that the C allele is associated with higher expression of gene A. So we can assume that gene A in patient has a higher expression than in controls. So we can conclude that the expression of gene A is associated with the disease. An important point is that this association is more likely driven by a genetic factor. So in the best case scenario, it's not a consequence of the disease, as, for example, we can find in RNA profiling studies. But in this case, there is the SNP acting on the gene expression. So we can conclude that the expression is associated. There is a causal relationship. This is a typical workflow. There are a couple of softwares. I like to use Fusion for this type of analysis. We just need the summary statistics. So because they are, it's more easy to access to them online instead of the raw data. We need to clean the summary statistics. So for example, compute the zeta, that's the zeta score, it's the effect size we use during the TWAS, remove duplicate SNPs, remove strand inconsistencies. And then we need expression weight. We can find, if we use Fusion, we can find these pre-computed weights from GTX, so we don't need to do additional analysis, but we can compute also our weight starting from some other data set. We do the expression imputation, of course, this is oversimplified, and we run the gene disease association with different models. And we adjust the result for multiple testing. Usually we adjust the result accounting for the number of genes tested and the tissues tested according to our hypothesis. And finally, this is the last one. We have the result visualization. Usually we use the Miami plot. So we have the difference with the Manhattan plot is that we have two directions of effect. We have the zeta score. The zeta score is the main effect size we have from the TWAS. And if it's positive, an increase of expression is associated with the disease. If it's negative, a decrease in expression is associated with the disease. These are a couple of useful resources. So there are a couple of papers for GWAS, TWAS, and the main softwares we use. I suggest to use, but there are many others for this type of analysis. So I'd like to thank the organizer for this webinar, all the panelists. There is an Alzheimer Consortium, the ADCC grant, and the ADHS grant. And for questions, you can also write me an email. Thank you very much. I will take your questions.
Thank you, Dr. Perez. I'm going to hand it over to Dr. Chen for some final words.
Thank you for all the speakers and for the time we have. We won't be able to have any question for the last speaker, Dr. Perez. Sorry about that. But I want to say thank you to NAC and Data Core Steering Committee, especially Heather and Hannes for all their working with us in the past several months to have this webinar prepared. They gave a lot of constructions and very helpful hint for making this successful. Thank you very much again. And also I want to convey the congratulations to every speaker from Dr. Acknowledgements. I'm going to end the webinar today for now. Thank you.